Our next speaker is Tim Sackton.
Hi, I'd like to start by thanking the organizers for giving me this opportunity to present some work I've been doing for the past couple of years on the convergent evolution of flightless birds. And the birds in particular that I've been working on are ratites, which are the widely distributed, large-bodied flightless birds that include ostriches, cassowaries, emus, rheas, and kiwis, which together with the volant tinamous make up the paleognath, which are one of the two basal splits within birds. Now, what's particularly interesting among many things about the ratites is that although historically they were considered a monophyletic group, recent in the past decade or so molecular evidence has very strongly supported ratite paraphyly, with these volant tinamous being nested within the flightless ratites. And so this implies multiple losses of flight within this group, and in particular makes flight loss within ratites a very good example of phenotypic convergence. So I'm particularly interested in the genetic basis of phenotypic convergence, and particularly whether we observe similar genetic changes occurring that are associated with these phenotypic convergences, or whether evolution uses independent paths to achieve the same phenotypes in different species. And so the ratites make a very nice system to look at this, and with this associated with this very large morphological change, the loss of flight. So in order to do this, we sequenced a lot of new genomes. So I'm showing you this is a phylogeny of the ratites. So here are the kiwis, the emu and cassowary, the rheas, and then here are the volant tinamous, the extinct moa, the ostrich, and then all of other birds are out here. And we've sequenced all of the genomes that have the little green flags are the new ones that we've sequenced. I'm not going to talk about the details in the interest of time. But combination of the phylogeny and the biogeographic evidence and the dating of these nodes has led us and other groups to suggest that flight has been lost perhaps 5 or maybe even 6 times if you include the extinct elephant birds, which are actually not in this phylogeny because we don't have nuclear sequence from them, but actually most closely related to kiwis. And so I've been using the assemblies and annotations that we've produced for this group to begin to get at this question of the genetic basis of convergence. So to start with, the very first thing I looked at was something that's been shown in some candidate gene studies, and I'm showing you an example here from Peter Endelfada's work on the evolution of cardenolide resistance in different insects. And what you see in this case is, from these candidate gene studies, is clear evidence for sequence level conservation, where the same amino acid mutation has occurred in the same gene in multiple independent lineages that's associated with this kind of resistance. So the very first question we asked with this data is whether you see evidence that this kind of process is sufficiently common to produce an excess of these kinds of convergent amino acid substitutions at a genome-wide scale. And so to do that, so this is what we're testing for, excess amino acid substitutions. To do that, and I'm showing you here just a subset of the bird phylogeny that we've used to do this for clarity, but it includes a total of about 40 species, including the 11 new paleognaths we sequenced. And so, of course, the lineages that we're interested in here are the ones shown in red, which are the ratites. And so what we like to do is estimate, for example, for all ratite-ratite comparisons, the number of times we observe the same amino acid substitution occurring at the same position in a protein, and then compare that to what we see for ratite-volant comparisons or for volant-volant comparisons. And if this kind of convergent process is sufficiently common to produce a genome-wide bias, what we would expect to see is that the ratite-ratite comparisons would be somewhere up here, and then the other comparisons would be somewhat down here. So we expect the number of convergent substitutions to increase with the number of non-convergent substitutions we observe in a protein, but we would expect the ratite-ratite ones to be somewhere above the line of the non-ratite comparisons. So that is actually emphatically not what we see. We see no evidence that this convergent amino acid substitution process is happening sufficiently frequently at a genome-wide scale to perturb the sort of the substitution process that we see. So the ratite points are shown in red here. And in fact, really all that's going on seemingly here is a pretty strong linear relationship between the number of convergent and the number of divergent substitutions. So I think that suggests, and perhaps this is not too surprising, that there's relatively little evidence for this kind of very specific sequence level conservation at a genome-wide scale in these organisms. And while this has been shown in several different candidate gene studies, it may not be particularly a strong enough process or a frequent enough process to impact genome-wide studies. Another thing that is commonly observed in candidate gene studies is the scenario where the same gene has convergently evolved in multiple independent lineages, but the identity of the mutations are not the same. So this is the example of MC1R. I'm showing you a figure from one of Erica Rosenblum's papers on the white sands lizards, but MC1R is commonly targeted to evolve in all sorts of different systems where you see these color morphs. So we don't have functional information. We don't know exactly what genes might be involved in this convergent flight loss, so we can't do quite the same test. So instead, what we're going to look for to try and get at this question of gene-level convergence is rate shifts in ratites. And this is motivated by some methods that Nathan Clark has developed, in particular a recent paper of his in MBE, which was just published, looking at these kinds of shifts in rates. So in this case, we're asking whether, again, all of these red branches are showing a convergent acceleration or deceleration in rates of protein evolution relative to the non-flightless lineages in black. And so this method is, the way I've done it is inspired by, but not exactly identical to what Nathan Clark has done. But so this is a figure from his paper, just kind of showing you the way this data looks when you do see evidence for convergence. So in blue, so this is looking at marine mammals. In blue, you see the distribution of a sort of relative measure of protein evolutionary rate for the non-convergent lineages. And then in red, you see the same measure for the convergent lineages. And you can see that all of these points in red are shifted to the right, suggesting that there's been a convergent acceleration of the evolution of this protein in these mammals. So we're going to do the same thing in ratites. So this is basically the idea. This would be the distribution of a normalized measure of protein evolution. On the non-ratite lineages, the red dots will show the ratites. And this is what we would see in a case where we see no evidence for a convergent rate shift. If instead we see, like in this NAT3 example from Nathan Clark's paper, that the normalized measure of protein evolution for the ratite lineage is shifted to the right, that would be evidence for convergent accelerations of that particular protein. So what I'm going to show you on the next slide is just a histogram of the p-values from running this test on every single gene in the genome for which we have a good alignment. And what you can see is that there's essentially no evidence for rate shifts, convergent rate shifts in the ratites. So this distribution is flat. If you do any kind of multiple test correction, there are no significantly convergently accelerated genes in ratites. So again, I would argue this is evidence that we see essentially no evidence for this kind of gene-level conversion, at least sufficiently common to impact things on a genome-wide scale. That doesn't rule out the possibility that there are still specific mutations that are important. So if you were at David Kingsley's talk last night, this may actually not be too surprising because we know from his work and others that a lot of convergent evolution, particularly of morphology, is associated with regulatory variation. So of course that we really wanted to get at that, and that is the next thing we looked at. So in order to get at regulatory variation, we don't have a well-characterized set of enhancers. So instead we used an approach from that's been developed in the sort of Adam Siepel and David Hauser labs to look at these conserved non-coding elements or non-exonic elements. And so in order to do this, we produced an alignment of 42 species of birds and non-avian reptiles. So this includes these neognaths, also the ratites, so kiwis, emus, cassowaries, rheas, and ostrich, the volant tinamous, which are paraphyletic within them, and then some non-avian reptile outgroups. And using this alignment, we annotated about 284,000 conserved non-exonic elements, which we consider to be putative, although for sure not definite enhancers. And then what we did is for each of these ratite clades independently, we test using Phylogenetic, the part of the program that Adam Siepel developed, whether we've seen a loss of conservation specifically on this lineage in the face of maintained conservation in the volant birds. So we allow the reptiles to also not be conserved for that sequence. And then we can do this for each of these 284,000 CNEs and then ask whether we see any individual CNEs that show multiple losses of conservation on different ratite lineages. And so when we do that, we actually see something pretty interesting. There are 6 of these CNEEs that are lost on all 4 ratite lineages, and more that are lost on either 2 or 3. And there's quite a number that are lost on at least 1. So before we get too excited about this, though, we want to make sure that this is not attributable to one of 2 possible alternative explanations besides these are regulatory elements that are involved in the loss of flight. So one possibility is that just by chance, if we imagine a sort of stochastic model where CNEEs are sort of turning over sort of by chance on each lineage, we might expect to see some number of CNEEs that are convergently lost on multiple lineages without that necessarily meaning that there's some sort of non-independence going on. And so in order to test that, what we do is we just do random permutations where we just randomly take the number of CNEs that we observe that are accelerated on each lineage, assign them at random, and see what we get. And so what you see here is the null distribution for the number of CNEs that we'd expect to observe to be accelerated in at least 2 clades just by chance alone based on the number of CNEs we observe accelerated in each independent lineage. And the red line is what we see in the real data, so you don't really need to do statistics to say that this is incompatible with that null model. That is true also if we use this 3-clade or the more stringent 4-clade example where we never, in fact, see all 4 clades lost just by chance. The other possibility, of course, is that this does represent some non-independence here, but it's not necessarily specific to flight loss. Maybe any random pair of species we looked at or set of 4 species we looked at would show evidence for these convergent losses. So in order to test that, what we did is I just picked sort of at semi-random a set of neognath lineages that are as equivalent as we can make it to the ratite phylogeny and ask how much convergence do we see in those lineages where there is no phenotypic convergence that we would expect would drive this process. When we do that, what you see, so what I'm showing you here is the proportion of convergently accelerated CNEs relative to the total number of accelerated CNEs across all lineages, for in red the ratites, and in blue the sort of random set of neognaths. And what you can see is you never see in our sort of random set of neognaths either 3 or 4 clades with this evidence for convergent acceleration. You see a much lower fraction with 2. And so this, I think, suggests that there's actually a biologically real excess of these convergently accelerated CNEs in the ratites and makes them particularly interesting as candidates to look at for perhaps being associated enhancers involved in limb development or something like that could be related to flight loss. Okay. This is my last slide. So the process that I think is going on here then is from this Volan ancestor, we have these multiple independent losses of flight. And in each of these cases, there's a set of putative enhancers that are convergently lost. So we see very little evidence for genome-wide convergence in the protein coding genes. But as I just said, there's a subset of these putative regulatory elements that have repeatedly experienced changes in evolutionary constraint. And of course, the next thing that we want to do is test these functionally. So currently, what we're working on in this project is trying to develop assays in chickens to take the chicken version of each of these CNEEs and the ostrich or emu or cassowary version and see whether they actually drive differences in expression. And so with that, I'd just like to acknowledge Team Ratite. So this project has been led by Scott Edwards at Harvard and members of his lab and some other PIs and funded by NSF. And I will now stop and take questions. Thank you.
Very nice work. I'm just going to ask the obvious question is like, if you look at what genes are close to your rapidly evolving enhancers or putative enhancers, what genes are those or what are the enrichments?
Yeah, so for those, we've looked most carefully at those 6 putative enhancers that seem to be lost conservation in all 4 ratites, and 4 of them actually are genes that are expressed in the developing limbs of chickens. So that's potentially very exciting. But as of yet, we don't have evidence that the ratite, we're working on it, but we don't yet have evidence that the ratite-specific versions have changed how they drive expression.
I feel like I missed the explanation of why you can rule out the possibility that the ratites had bottlenecks or reduced population size? And so they would suffer just much greater just rate of loss of function?
So if all of the ratite lineages had had a bottleneck and so had lost these things. I mean, I guess that's possible. These are elements that seem to be conserved in every other bird, so they're probably doing something important.
Do you know about their current effective size? Are they reduced?
It depends on the species. I mean, kiwi, we can talk about it. Kiwis have a low population size, but something like ostrich, not necessarily. Okay, thanks.